So this week we're going to focus on just chapter 14, and this is going to cover the basics of inheritance, so how genes are passed on from one to the next, and we're going to use Punnett squares to look at how genes are inherited. The principles of genetic inheritance were discovered by a monk named Gregor Mendel, and he did this in a garden with pea plants where he looked at specific traits and just looked at many plants over many generations. And when you think about kind of the tools that he had available to him, it's amazing with the principles that he was able to deduce from just breeding plants for many generations. So here are some terms that we need to clarify. A character is some kind of heritable feature. In the plants, Mendel looked at several different characters. Character could be the flower color. Is the plant tall? A character can have many different variants. So each variant is called a trait. So if the character is flower color, there are several different traits. So it could be a purple flower or a white flower. So Mendel was able to track the occurrence of these specific traits over many different generations. What he did was he chose characters that had only two distinct forms, so tall, short, purple, or white. Mendel started with plants that are called true breeding. A plant has a specific trait, and they pollinate with themselves. Remember, plants can reproduce with themselves, that they're always going to produce that same specific trait. So that trait breeds true. For example, a white flower plant breeds with a white flower plant, and all of the offspring are white flower plants. So that plant would be considered true breeding for white flowers. So here's Mendel's experiment, and we'll just use one example of flower color, but he did this for 8 different characters. In this case, we have a true breeding purple flower plant and a true breeding white flower plant. So both of those, if they mated with themselves, would produce the same color. So Mendel wanted to see what happened when I take these 2 true breeding varieties and mate them together. This is called the P generation for the parent generation. The P generation is always going to be true breeding parents. So what happened when he did this was he found that the next generation, this is called F1 for filial 1, meaning this is the first generation after the parents. These were all purple. So for some reason, the purple completely covered up the white, and there were no white flowers in the second generation. We know now that these individuals were hybrids. At the time, Mendel didn't know that. We know that they have one allele for purple and one allele for white, and that the purple was dominant, and so it covered up the white allele. Mendel didn't know this. So Mendel had this question at this point: is the white gone? Did purple completely erase it? If I mate this plant again, will I see white? What happened to the white? The next generation is called the F2 for filial 2, where he mated this with itself. So this is self or cross-pollination between all of these individuals. So it's mating either with itself or with its siblings. And what he found was that most of the third generation here was purple. So this is about 3/4 purple plants. But the white reappeared. We had about 1/4 white plants. So now Mendel knew that the white didn't get lost. However, for some reason in the second generation, that F1 generation, the white flowers got completely covered up. And then they reappeared in the third generation, the F2 generation. So what he deduced from this is that the purple flower color was dominant, meaning that it dominated over the white. So when they were both available, purple was chosen. And the white flower color is a recessive trait, meaning that it is completely covered up by the purple when both of them are present. What he found by doing the F2 generation was that it was not diluted or destroyed. The white flowers came back as fully white flowers. So it was either purple or white. There was no mixing or blending or dilution of these traits. So Mendel decided that there must be some heritable factor that delivered the trait either in its entirety or not at all. And so what he was considering a heritable factor is now what we call a gene. So this gene is a sequence of DNA, and now we know that the gene would either code for purple flowers or white flowers. And if they had the gene for both, if they had one gene for purple and one gene for white, because purple was dominant, it would completely cover up the white, and you would get purple flowers. So now we're going to go through the many contributions Mendel made. So these are the conclusions that Mendel came to as a result of his experiments. So the first conclusion was that alternate versions of genes account for the variations in inherited characters. This means that that heritable factor, the genetic factor, is what causes the different traits, whether it's purple or white. So we call these different versions of the genes alleles, and we usually represent them with a letter. The letter is usually going to correspond to the dominant gene. So if we're talking about purple versus white flowers, usually we'll use a capital letter P to indicate the dominant allele. And we use P because the dominant was purple. And then we keep the letter the same to make sure that we know that we're talking about the same character. And we might use a lowercase p to talk about the recessive allele. If an individual has a dominant P, this is a trait that codes for purple, or the dominant trait. And then an individual with a lowercase p will code for the recessive trait, the white flowers. He also decided that these genes have a specific place on the chromosome. So if this is the chromosome, the gene will always be in a specific location or locus. So a locus is just a specific location on a specific chromosome. The second discovery that Mendel made was that for every character, so for example, flower color, an organism gets two alleles. We get two letters or two versions of the gene, one from each parent. So in the case of flower color, each plant gets one allele for flower color from its mother, and it gets one allele for flower color from its father. And then the combination of those two alleles will determine what flower color that plant actually has. So sticking with our flower color example, there are three options. You can be capital P, meaning that you got the purple color allele from both parents. Or you can be little p, meaning that you got the white allele from both parents. These are both considered homozygotes. This word homo here means same. So in this case, these both have the same letter casing. So big P, those are both capital. Little p, those are both lowercase. So this individual would be called homozygous dominant. And this one would be homozygous recessive. There's one more option, and the other option is that you can have both, a big P and a little p. These organisms are what we call the hybrids. So these are going to have two different alleles. So these are called heterozygotes. Hetero here means different. Another term for these is hybrid. So that entire F1 generation were heterozygous. So these are heterozygous. The homozygous individuals can also be called true breeding. So we know that all of the true breeding parents in the first generation were all homozygous, and that's why they produced all flowers of the same color. And then all of the individuals in the F1 generations were hybrids because they had one big P from one parent and one little p from another parent. These are what we call genotypes. This is the actual genetic sequences that each individual carries in its chromosomes. The third conclusion that Mendel came to is that when the two alleles at a locus differ, so specifically talking about heterozygous, the dominant allele, big P, determines the organism's appearance, and the other allele, the recessive, the little p, has no noticeable effect. So this is called complete dominance. So this is a heterozygous individual. We can use the genotypes to figure out the physical expression of these genes. What color are the flowers? The phenotype is the physical expression, whereas the genotype is the alleles. So an individual that has big P, those are both dominant for purple flowers. The phenotype is the actual trait. This plant is going to have a purple flowers. So that's the physical expression. The other homozygous little p, those are both recessive. So those are both going to code for white flowers. So it makes sense that this plant would end up with white flowers. So what this third conclusion says is that when an individual is big P, little p, meaning that it has one capital and one lowercase, one dominant, one recessive, it's actually going to have purple flowers. It's not going to have any white. It's not going to be like diluted or mixed. The dominant purple trait completely covers up the recessive white trait. And so often when we talk about, this is called complete dominance, or sometimes we equate this to the term called Mendelian genetics. The fourth main conclusion that Mendel made is actually now got its own name. So this is called the law of segregation. This is an important one for understanding how Punnett squares actually work. So what it says is that the alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. So this is happening during meiosis. So here I have a diploid cell, and this individual, I'm going to use big P, little p, just to say that's the genotype of this individual. Diploid means that I have both sets of chromosomes, right? So they have one chromosome here, another chromosome here on the homologous chromosomes. Separation of homologous chromosomes during meiosis. So we're going to do meiosis, and we're going to end up with gametes. This individual can pass on eggs or sperm that have half of the number of chromosomes. So right now we're just focusing on these two chromosomes right here. The two options for the gametes, let's say this is an egg. The egg could be big P, or we can make eggs with little p. You're not going to make eggs with both of them in there. So this is the idea that during meiosis, these two are going to separate from each other, and they're going to be inherited independently, or they're going to separate. And so we end up with gametes with only one allele for each character. So this individual can pass on a dominant allele or a recessive allele, but not both. We've already seen that the genotype is the actual alleles, the genetic makeup, and the phenotype is the physical appearance. And we can always go from genotype to phenotype. So given the genotype of an organism, it's easy to decide what the phenotype is going to be because we know the genetics. You cannot always go from phenotype to genotype. It's hard to distinguish what the genotype of a plant is from just the phenotype. So if the phenotype in this example is white, we can figure out the genotype. When we're looking at the recessive trait, you are going to be able to figure out the genotype. You know that it has to be little p, little p. That's the only way to get white flowers. However, this is an example. This is the F1 or the F2 generation here, where we got 3 purples to every 1 white. We have 3 purple, which is the dominant trait. These actually have 2 different possible genotypes. They can be big P, or big P, little p. You cannot look at a purple flower and determine its genotype from just the color of a dominant trait. So when you have a recessive trait, yes, you can figure out the genotype because there's only one possibility. However, when you look at a dominant phenotype, like purple, you have 2 different options for what its genotype could be. So we use a tool called a Punnett square for determining the ratio or the genetic possibilities of offspring in a cross. So let's look at Mendel's experiment and show you the Punnett squares for the different generations. In the P generation, Mendel started out with 2 true breeding plants. And we know that one of them was true breeding for purple flowers, so that is big P. And they mated that with another individual that is true breeding for white flowers, so little p, little p. And I like to just draw the little tail on there to help me distinguish these letters from each other. Next, we know the law of segregation. Each of these plants can only pass on one allele at a time. So this parent can pass on, let's say this is mom, an egg with a big P or an egg with a big P. This parent, let's say it's dad, can pass on a sperm with a little p or a sperm with a little p. Those are the same because they're true breeding. So what we do is we're going to line up a Punnett square. So a Punnett square is going to take the gametes, not the parent genotype, the gametes, and arrange them in all possible combinations. So I have 2 gametes from mom, 2 gametes from dad, so I need a 2 by 2 Punnett square. So I will take the mom's gametes and put them over here, and dad's gametes and put them over here. Then all we have to do is combine all of the possible combinations, and we just drag them across and drag them down. So big P, little p, big P from mom, little p from dad, big P from mom, little p from dad, big P from mom, little p from dad. And just a convention, you always draw the capital letter first and then the lowercase letter. It doesn't actually matter what order they're in, but to make it simpler, just do capitals first and then lowercase. So now we can see that these individuals make up the F1 generation. So if you remember what Mendel saw, is he saw that the F1 generation was 100% purple flowers. And so here we can see that represented in the genotypes. We've got all 4, so 4 out of 4 are big P, little p. So they're all heterozygous or hybrid. And in here we can see the big P completely covers up the little p, and we get all purple. So then he did another cross. Mendel mated these individuals with each other. So next we have the F1 cross, where he mated an F1 individual, which is big P, little p, with their sibling, big P, little p. Remember, this is plants, so that's okay. So again, we have the law of segregation. This parent, let's say mom, can make an egg with a big P or an egg with a little p. And then this parent is dad, can make a sperm with a big P or a sperm with a little p. And then each one of them will pass on one allele. So we have 2 possible alleles, or 2 possible gametes from mom, 2 possible gametes from dad. So we need a 2 by 2 box. So let's put mom over here on the side where we've got big P or little p. Dad we put over here on the top, and it doesn't matter which one goes on which side or the top. And then again, we're going to drag them over and down. So in this square, we will get big P, big P from mom, big P from dad. In this square, again, we get big P from mom, little p from dad. In this one, we get big P from dad, little p from mom. It doesn't matter where they came from, just capitals first, then lowercase. And then in square, we get little p from mom, little p from dad. So this box here represents the F2 generation, where Mendel saw we had 3 purple to every 1 white flower. And so what we can see is the big P, this is going to be a purple flower. Big P, another purple flower. Big P, another purple flower. So we get 3 out of 4 are purple, with either big P or big P, little p. And then we have this last one here, little p is the 1 out of 4 that is white with little p. Oftentimes we summarize these Punnett squares with the genotype ratio. And so a genotype, again, is looking at the alleles, where we have 1 big P for every 2 big P, little p's, for every 1 little p. So this is one way to kind of describe what's happening here. And then the other description we often use is the phenotype ratio, which again, phenotype is the color, where we would have 3 purple for every 1 white. So you should expect to be able to get questions where you have to draw a Punnett square and then describe the ratio of the offspring, either their genotypes or their phenotypes, like this. What we saw in the F1 offspring there is that the cross was a monohybrid cross. Mono means that we're looking at one character. So we're only using one set of alleles for each parent. And hybrid, of course, means that they're heterozygous. So when we had big P, little p, crossed with big P, little p, this is one character. We're just looking at flower color. And both parents are hybrids, where they have one big, one little, one big, one little. And so a monohybrid cross is always going to give you a 3-to-1 phenotype ratio and a 1-to-2-to-1 genotype. So let's go back to the question of, I hand you a purple flower. And the question I have to ask you is, what is the genotype? Well, you don't know. From just handing you a purple flower, it could be either big P, or it could be big P, little p. Both of those will give you the exact same purple flower. So in order to figure out what the genotypes were of his F2 cross, because Mendel didn't know whether the purple flowers were big P or big P, little p, he did a test cross. So a test cross is when you're going to take your purple flower and you're going to mate it with a homozygous recessive individual. So I'm going to mate this with a true breeding white or little p individual. So let's go through an example of a test cross. There are two options. I handed you a purple flower. It is either big P, or it's big P, little p. One of those two is true. So what I'm going to do is I'm going to mate it with a white flower. So I'm going to draw two crosses here to show you the two options. And then based on what you would physically see, you would be able to decide which parent you had. This individual can make alleles or gametes of big P. This individual can create only little p or little p, right, sperm. We'll go ahead and draw our 2x2 Punnett square. So big P, little p, and then fill in the square. And this should look just like the P generation cross from Mendel. And the result of this is we will get 100% purple flowers. So if you did this test cross and got 1,000 individuals, you would expect all of them to be purple. The other option for your plant was that it was big P, little p, in which case mom here could make a big P or a little p, and dad could make a little p or a little p. So then you can draw the Punnett square, 2x2. Here's mom, big P, little p. Here's dad's gametes, little p. So then we combine them, and you can see that they make 2 heterozygous and 2 homozygous recessive. So from this, we would expect 50% to be purple and hybrid, big P, little p's, and 50% to be white, little p. So if you did this cross and got 1,000 offspring, you would expect about half of them to be purple and about half of them to be white. So what you would do is you would then observe the offspring of this cross, and if you saw any white flowers, and it should be about half, you would have to conclude that your parent was big P, little p. And if you saw no white flowers, you would conclude that your parent was big P. The last law that Mendel described was the law of independent assortment. And this says that every pair of alleles segregates independently of any other pair of alleles. So now in this case, we're going to look at 2 different characters. We'll keep our flower color, which is P, and then we're going to look at another character, let's say maybe plant height. So in plant height, a tall flower is dominant, and a short flower, we'll use T again, would be recessive. What Mendel's law here says is that the alleles for plant flower color, P, and the alleles for plant height, T, they don't depend on each other at all. P will assort by itself randomly, and T will assort by itself randomly. So they're not linked to each other, they don't depend on each other at all. The way that Mendel showed this is he used what's called a dihybrid cross. Di means 2. We're going to be looking at 2 characters. We've got flower color and plant height. And hybrid means that both parents are hybrids for both traits. So the parents are going to be big P, little p, big T, little t. That is a dihybrid individual. They have two traits, P and T, and they are both hybrid, heterozygous. So let's look at an example of a dihybrid cross. So we are going to take two dihybrid individuals, big P, big T, little t, and cross them, big P, big T, little t. The first step is always figuring out what are going to be the gametes. Before this was pretty simple, you just took the two letters and separated them. Now we have to make all possible combination of gametes. Remember that a gamete will have exactly half of the traits. But you don't want a gamete to have two P's or two T's. Each gamete needs to have a P and a T, because they're going to pass on one allele for each of these two characters. We can make all of the different possible combinations. The trick I like to use, if you've been in algebra before, is called FOIL, to figure out the 4 possible gamete combinations. These both parents have the same genotype, so they're going to have the same gametes. So I'm just going to draw this here, big P, T, little t. So F stands for first. So that is going to be the first of each character. So the first is going to be big P and big T. So big P, big T. O stands for outside. So those are the two on the outside. So the outside is going to be big P and little t. So that's going to be big P, little t. And each of these is a gamete. Like those might be an egg. I stands for inside. So those are the two on the inside, the little p and the big T. So the next egg would be little p, big T. And then L stands for last. So that's the last one in each pair. So little p, little t. So with 4 different alleles here, there are 4 possible combinations of gametes. So these are the gametes. Each parent is going to be able to make the same 4 gametes because they have the same genotype. So if this is mom, she can make 4 eggs. Big P, big T, big P, little t, big T, or little p, little t. Dad can make the same 4 sperm. Big P, big T, big P, little p, big T, little p, little t. So now I'm looking at the number of gametes, and I have 4 from mom and 4 from dad, and that's more than a 2x2 square. I'm going to need a 4x4 square for my Punnett square. So then we do the same thing as a normal Punnett square, and we're going to take the gametes and put them on the sides. So I'll put mom on the side here, big P, big T. They go together. Remember, this is one gamete. Big P, little p, big T, little p, little t. And then dad goes on the top, big P, big T, big P, little p, big T, little p, little t. Each gamete gets its own row and its own column. You can do a quick check to make sure that you're doing this right. Each of these parents has 4 genes or 4 chromosomes. This is the 2N diploid status. And each of these gametes here has 2 chromosomes, 1P and 1T, which is the N haploid gamete. And then when we combine them back into an offspring, each of these boxes represents an offspring. This is going to be a zygote. That's a diploid cell. So you combine the same way across and down. This individual should have 4 letters. Big P from mom, big P from dad, big T from mom, big T from dad. The convention is to put like letters together and then always capital before lowercase. So we put the P's together and the T's together. That's going to make it much easier for you to read what they are. So now you can check your Punnett square against mine to see if it matches. You should get the exact same Punnett square. When we describe these offspring, there are many different genotypes. We don't usually talk about the genotype ratio of a dihybrid cross. However, there are 4 possible phenotypes. A plant can be purple, and tall. Those are the two dominant traits. A plant can be purple and short, so purple being dominant, short being recessive. A plant can be white and tall, or a plant can be white and short. So when we look through this Punnett square, we have a total of 16 boxes, which means that if we count each of these different characters, we should come up with 16 as the total number. So let's start with finding all of the plants that are purple and tall. This means that they have a capital P and a capital T. They can be homozygous dominant for both, like this one here, big P, big T's, or they could be heterozygous. This one's purple and tall. Purple, tall, purple, tall, tall. This one here is purple, but it's short. This one's purple and tall. This one also is purple and short. This one's purple and tall, purple and tall. This one is tall, but not purple. Same with this one, tall, but not purple. Purple, tall. This one is not tall. This one is not purple, and this one is not purple. So you can see that I've crossed out all of the ones that are purple and tall. So now I can count them. 1, 2, 3, 4, 5, 6, 7, 8, 9. There should be 9 that have dominant phenotypes. Now let's count all of the ones that are purple but short. So here we've got purple and short, purple and short. This one's white and tall. This one's white and tall. This one is purple and short. Notice that I am not counting any of the ones that I already crossed off. Once it has been accounted for, you do not count for that individual twice. The rest of these are going to be white. So there are 3 purple and short. Now let's find all of the ones that are white but tall. So we've got little p is white, and then all it needs is 1 capital T. So this one is white and tall, white and tall, white and tall. This one's going to be white and short. So we're going to end up with 3 white and tall, and then this last one is white and short. So the typical phenotype ratio is 9 to 3 to 3 to 1. So every time you see a Mendelian dihybrid cross, you should expect 9 that are both dominant. These are dominant. 3 that are dominant and recessive. 3 that are recessive and dominant. And then 1 that is both recessives. Complete dominance is the Mendelian genetics, where when we had big P, little p, the big P dominant covers up the little p for white, and we end up with purple-only flowers. There are exceptions to these rules of dominance that we call non-Mendelian or exceptions. And first one is called incomplete dominance, and this is going to happen in a hybrid where we have kind of blending. So let's say we have snapdragons where big R is dominant for red, little r is recessive for white. If this was completely dominant, this flower would be red. However, because it is incomplete dominant, we get mixing. So this ends up being a pink flower. It's just like if you took 2 paints, right? A red paint and a white paint, and you mixed them together, you would get a pink color. And so it's a blending or an intermediate between the 2 phenotypes. Codominance is cooperative, meaning together. This is when you have 2 different dominant alleles. So, for example, we can have an allele for a brown cow or a dominant allele for a white cow. Notice these are different letters because they're both dominant, so they are different. In a hybrid where we have RW, instead of getting a light brown cow, what you get is a white and brown spotted cow. So the 2 alleles are both separate. You can see patches of white and patches of brown, and they're distinguishable. The colors are very distinct. You can physically tell me which, if I plucked a single hair from the cow, I would be able to tell if it was white or brown. So it's not a mixing, but what it is both alleles being expressed separately. So here's an example of incomplete dominance using snapdragons. So the way that we write these alleles is not always just one letter. So we write C to represent that it stands for the flower color, and then the superscript R tells me whether the allele is dominant or recessive. So for the P generation, we have two true breeding flowers, CR, CR, CW, CW. They can each make gametes. So here's the gametes. This parent can make CR gametes, and this parent can make CW gametes. They've only written one of them here. You could imagine just having another one next to it to show that there's two gametes from each parent. Next, we have the F1 generation. So we're going to see that all of these individuals have one gamete from each parent, giving them the hybrid CR-CW alleles. And so we're going to see 100% pink. This, in the Mendelian, we saw 100% purple flowers because big P covered up little p. In this case, instead of one covering the other, they blend together, and we get all of them having this kind of intermediate mixed phenotype. Then we can do the F2 generation, where we breed these two together. So we're going to do CR-CW crossed with CR-CW to get the F2 generation. And you get 1 out of 4. So 1/4 is CR-CR, which will be red. There's 2, so 2 out of 4, which is going to be 1/2, CR-CW, which will be pink. And then again, 1 out of 4, which will be CW-CW, which will give me white. So for incomplete dominance, the genotype ratio is the same as the monohybrid cross, 1 to 2 to 1. But the phenotype ratio is now going to be the same as the genotype ratio, where we have 1 red to 2 pink to 1 white. So this is red, pink, white. Just because something is dominant does not mean it is common. Most of the time, something that's dominant is common, but that is not necessarily true. For example, polydactyly is having many fingers. So this would be having 6 or more fingers on your hands or your toes. This is dominant. I want you to think about how many people you actually know that have extra fingers or toes. The statistics are 1 out of 400. That's not very many. So even though something is a dominant trait, doesn't mean that it is necessarily common. Another difference from Mendelian genetics is the ability to have multiple alleles. So before, when we talked about alleles, there was a capital letter and a lowercase letter, and those were the two options. We had big P or little p. Some traits have more than one possible alleles. So for example, we've got human ABO blood types. So there are 3 possible alleles. We use the letter I to represent whether it's dominant or recessive. So IA is going to be the A allele. IB is dominant for B type blood. And then little I is recessive, and this is going to code for O type blood. So this is the ABO blood grouping. And there's lots of different ways to combine these 3 alleles. So if we have IAIA or big IA, little I, we have a homozygous dominant and a homozygous recessive that gives us an individual with the phenotype of type A blood. So what we've got here is the physical representation is that it's got these type A spikes on the blood cell. So those are called antigens. And so a person with type A would have type A antigens on their blood cell. A person with type B, there's 2 possible genotypes, big IB, or big IB, little I-O, meaning that they are homozygous dominant or heterozygous here. So A is dominant to O and B is dominant to O, so we get complete dominance in these 2 cases. So both of these individuals would be type B, and you can see they drew the type B antigens with circles instead of triangles. this is an example of co-dominance. We have big IA and big IB. Those are both dominant alleles. They are equally dominant to each other. So instead of having some kind of blending, what happens is this blood cell actually has both triangles and circles. So it represents type A and type B, so we call this type AB. And then the last option is if you have little I, you have no antigens on your blood cells, and that makes you type O, which is the universal donor because you don't have any antigens. So you can give blood to anybody without your blood clotting up or causing kinds of issues in like blood transfusions. Pleiotropy is when you have one gene that causes many effects. So these are often genetic mutations for hereditary diseases. So an example of this would be cystic fibrosis or sickle cell disease, where it's one change in the allele. However, it's going to cause many different effects systemically throughout the body. So that's pleiotropy. Epistasis is where one gene alters the expression of a different gene at a different spot. So let's say we have two different chromosomes. There's maybe a gene over here. So for example, in Labradors, we have a gene for pigment color. So this might be big B. And of course, you have two chromosomes with that gene on them. So maybe they're little b. And then they have a different chromosome with a different gene on it. And the other gene is going to code for whether they have coat color or not. So E. So this other gene, which determines whether pigment is deposited in the hair or not, is going to affect the expression of this gene. And so we have a gene in one spot affecting a gene in a different spot. Notice now that we have two different alleles that we're looking at. So we're going to be looking at a dihybrid cross here. So if we take two black Labradors, so we've got big B codes for black labs, little b codes for brown labs, big E codes for yes, you have color, and little e codes for no color. So if you cannot express your black or brown color, you end up as a yellow lab. So when we take the two heterozygotes, right, big B, little e, little e, and we do the cross, you can see that this dihybrid cross is done here for you. Let's look at each individual. All of the individuals that are black or brown have dominant E traits, meaning that they code for yes, they do have color. So we end up with 9 black labs and 3 brown labs. And then notice that there's 4 yellow labs. 3 of them code for black labs, and 1 of them codes for a brown lab. However, those traits do not get expressed in this Labrador because the other trait supersedes it. These are all little e, meaning that they code for no pigment in the fur. So what ends up happening is the fur gets made without the black or brown color, and you end up with 4 yellow labs. So in an epistasis, we see a slightly different phenotype ratio here from the typical dihybrid, where we have a 9:3:4, and it's just that that last 3 and 1 are combined into 4 here. These still all add up to 16. Another way that traits can be affected differently is by having polygenic inheritance. This is kind of the opposite of pleiotropy. So this is when many different genes come together to affect one character. So for example, human height is a good example. So you don't have one single height gene, otherwise we would all be either tall or short. Instead, we have many different genes throughout our body, over 180 genes, that affect your height. Things like growth factors, stuff like that. And so just because you have 2 really tall parents doesn't mean that you're going to be really tall. It's most likely. But there's a lot of different genes that can get affected. Human skin color is an example as well. So usually what we would see is from this one pairing, there are 3 different traits here. This is called a trihybrid cross here. And you can see there's a massive amount of offspring potential. So we have an 8 by 8 Punnett square here. we're going to see, you can see the colors shaded with the potential skin color of each offspring based on just which genes are dominant, which genes are recessive. So we start with 2 heterozygotes, and you can end up with somebody who's really, really light to somebody who's really, really dark. And a lot of these polygenic traits we tend to see on kind of a normal distribution, where there's most people in kind of the moderate, medium traits, where we have fewer people on the 2 extremes. So for example, height, if you looked at a whole bunch of people and looked at their height, most people would be in this general average height. You'd have very few really short people and very few really tall people. Genes do not determine every single effect on your phenotype. The environment that we live in does impact the way that we look. So this is kind of that whole nature versus nurture thing. So here's an example of hydrangeas, and these are the exact same hydrangeas with the exact same genetics. And they are grown in 2 different soils. This is going to have a high pH, so this is a basic soil. And then this is an acidic soil, so this would be a low pH soil. And you see that the flowers end up being different colors. So even though they have exact same genetics, they can end up with different colors. So many genes are combined with environmental influences, and we get these multifactorial results. So there are many traits that are just simple, like you're dominant or you're recessive, and that's it. But a lot of characters in human expression are dependent on multiple genes, as well as interactions with your diet or the environment that you live in, to give you different expression of different phenotypes. So we use a tool called a pedigree to track the relationship of traits across multiple generations, so from parents to children through many generations. So this is what a pedigree looks like, and it's basically just a family tree. So a square is a male, and a circle is a female. And then the shading makes a difference. So each pedigree that we will look at is going to be concerned with some trait. So if a block is shaded in, that means that they have the trait, whatever it is that we're looking at. In this specific pedigree, the trait that we're looking at is a widow's peak. Usually pedigrees are looked at to measure diseases through family trees. This one is just looking at a specific trait, which is the widow's peak. In this case, a widow's peak is a dominant trait. So anybody who's shaded in purple is either going to be a male with a widow's peak or a female with a widow's peak. And the widow's peak is the little peak there on the hairline on the forehead. Anybody who is not shaded in, so these are going to be white. The shaded colors can be any color. They're usually just black or white. This means that they do not have the trait. So this person would have no widow's peak. The line between 2 partners is called a mating or marriage line. So that indicates that this male and this female mated and had some offspring. And then these are their offspring, and they're always in the order of birth. So this is the first daughter, and then this is the second daughter. There are different kinds of genetic disorders that we use pedigrees to examine. So we'll talk about some recessive alleles here. So recessively inherited disorders only show up in individuals who are homozygous recessive. So in this case, the example down here is albinism. And albinism is a single allele recessive disorder. So you'd have to be little a to be albino. And albino means that you don't have pigmentation in your hair and your skin. So you can see in the picture that the girl on the right is albino. She doesn't have any pigmentation in the hair, whereas her sister here is normal and does have pigmentation in the hair and the skin. Recessive alleles can be carried through generations in people called carriers. So somebody who is big A for normal pigmentation and little a would be normal, right? This is a dominant recessive genetic situation, so big A would cover up little a, and this person would look normal. However, if somebody who is big A little a mates with somebody else who's big A little a, they have a 1/4 chance or 25% chance of having a baby who is albino. So most people who have recessive disorders are born to parents who are carriers. And that's usually because most people who have recessive disorders, a lot of recessive disorders can be extremely serious, and so those people don't tend to reproduce. Albinism is one of the ones that does not affect reproduction, so as long as you take measures to protect yourself from the sun, an albino person doesn't necessarily have any worse chance at reproducing. So in a different recessive allele, one that causes a disease, if the disease is very rare, it's unlikely that two carriers will meet because the frequency of that recessive allele in the population is so low. And so usually the frequency of recessive alleles and recessive diseases is fairly low. Consanguineous meeting, so con means close blood matings. So this would be matings between very close family members. Those tend to increase the chance of carriers meeting with other carriers and carrying the same rare alleles. So in our society, we call this incest, and we have taboos and laws against marriage of close relatives. Most animal societies actually have mechanisms in place to prevent close mating like this so that rare genetic disorders don't meet each other in close families. So this is kind of evolved in society as a natural selection against rare genetic disorders. Cystic fibrosis is another lethal genetic disorder. So this is a very common one. So this would be 1 out of every 2,500 people of European, of European descent. What happens with cystic fibrosis is the linings of the inside of the lungs and the digestive system become very thick. They get a buildup of chloride outside of the cells, and they're not able to absorb or transfer nutrients across those cells. And so people with cystic fibrosis often have issues with breathing in their lungs. One way to treat this is a lung transplant. And then sometimes they have issues with absorbing nutrients in the small intestine. And so this can be lethal if not treated. Another homozygous recessive disorder is called sickle cell disease. So sickle cell disease is about 1 out of every 400 people of African American descent. And what happens is the red blood cells, instead of being circular like they normally should be, end up being sickle-shaped. And these sickle-shaped cells can tend to build up on each other and cause clotting. And so this can get stuck in joints and cause pain. They're also not as good at absorbing oxygen and carrying oxygen. So somebody who is homozygous, so little r, would have sickle cell disease, and all of their cells would be sickled like this. Somebody who's a heterozygous, so big R, little r, says they have what's called sickle cell trait. So about half of their cells would be normal, and then another half would be sickle. And so they're usually healthy, but they might suffer some symptoms of sickle cell disease, but they generally live and are able to reproduce. And so this is how this trait continues to be carried on in the population, because carriers are okay, and then they pass that trait on. So if somebody who's a carrier mates with somebody else who's a carrier, they again have a 25% chance of getting someone who has the full-on sickle cell disease. So you would imagine that something like this would get selected out of the population. Eventually, the people who have sickle cell trait would be less fit to mate and reproduce, and then eventually these kind of diseases should go away. However, this is fairly common in African Americans, and it's 1 out of 400, and it's fairly common in areas in Africa where malaria is common. So you can see these two maps. This map on the left shows all the pink where malaria is common, so kind of western to central Africa here. And then over here is a little heat map showing the allele frequency of the sickle cell. So HS is the sickle cell. And so it's common also in kind of the same area. These really overlap here in this central western Africa. And the reason for that is because even though having sickle cell disease is damaging, in areas where malaria is common, having sickle cell trait, the heterozygous, so HSHS is what they're using here, is actually an advantage. This is something called heterozygote advantage. What happens is those people are generally okay. They might have a few symptoms, but they're generally healthy. And because some of their cell is sickled instead of regular, it actually helps to prevent them against malaria. Malaria, when it goes through a life cycle, has to actually reproduce in red blood cells, and it can't get into the sickled red blood cells. And so this is one of the reasons why the frequency of this disease is still very high, because having sickle cell trait makes you less likely to die of malaria. So sickle cell disease is not very common in first world westernized countries like the United States, where we don't have to deal with things like malaria. However, in Western and Central Africa, where malaria is still a huge problem, the frequency of sickle cell disease is actually much higher because it confers some advantage against dying of malaria. Other disorders can be inherited with dominant alleles. These are generally rare, and they usually arise by some kind of mutation. So, for example, achondroplasia is a form of dwarfism. There's several different types of dwarfism. This is just one of them specifically. And if you have capital D, because this is a dominant trait, this would be dwarfed. And this is a dominant disorder, so even somebody who only has one capital D would still have the dwarf trait, and then the only person who would be of normal height would be a heterozygote recessive. So that's what we mean here by it's a dominant disorder. Anybody who has a dominant allele is going to show that disorder. Typically, if a dominant disorder is lethal, that individual is not going to be able to reproduce and pass that trait on. That's another reason why dominant disorders are rare. Dwarfism, like achondroplasia, doesn't affect the ability to reproduce, and so that trait gets passed on because it's not lethal. Huntington's disease is a neurodegenerative disease, which over time prevents the brain from communicating with the muscles and actually degenerates neurons, and it's lethal. However, Huntington's doesn't set until about 30 to 40 years of age. So most people, by the time they're 30 to 40 years of age, have had the window of opportunity to already reproduce and pass that gene on to their offspring. So let's imagine if an individual finds out that they have Huntington's disease. So they would be big H, little h. It's not very likely that they'd be homozygous dominant. Let's say that they mated with somebody who did not have Huntington's disease. So little h. What is the chance that their offspring is going to also have Huntington's disease? So the chance that they have a baby, right, these are their offsprings, is actually 50% that they will have Huntington's, and only 50% that they'll be normal. Right? So little h is normal. Big H, little h means they'll have Huntington's. So dominant disorders have a high rate of transmission if they're able to be passed on. So it's a 50% chance. So somebody who finds out they have Huntington's would want to do genetic panels on any of their children to find out if they also have Huntington's. We do a lot of testing nowadays with modern medicine to look for genetic disorders so that parents are aware of their chances of passing on genetic traits, as well as prepared to deal with any genetic disorders that they find in infants. Most women who are pregnant will go through genetic screenings where they will actually look at the maternal blood, because some fetal cells do actually get into the maternal blood, and they can look for chromosomal disorders that way. If those tend to show that there's likely a genetic disorder, or somebody who's coming from a family that likely has genetic disorders, they might actually get more invasive testing. So amniocentesis is done a little earlier in the pregnancy, and this is where they would pull some of the liquid from the amniotic fluid, so that's the water that surrounds the fetus. And what happens is some of the fetal cells slough off, and they can do genetic testing on those to see if the baby has any genetic disorders. Chorionic villi sampling is actually taking a small piece of the placenta, and this again is going to have some fetal cells, and they can do the same kind of genetic testing. Both of those, while very informative, do carry a slight risk to miscarriage, to damage of the baby and the pregnancy. Most other techniques like ultrasound, maternal blood draw are usually just done visually in utero. When a baby is born, they take a ton of blood samples, and then they do tests for other genetic disorders at that point. And then there's other genetic disorders that are tested for throughout life as children age. Here I've got two examples of pedigrees, one of which is a dominant trait and one of which is a recessive. So I want to give you some practice here at recognizing dominant and recessive disorders. So the biggest thing to look for is carriers. A carrier will be somebody who does not show the trait, right? However, can pass it on to an offspring who does show the trait. So carriers are only going to occur in recessive genetic disorders. So if we look at the pedigree on the left, we see a male here who has the trait, who has passed it on to a daughter and a son. A female here who has this trait has passed it on to a daughter. And then these two both had the trait and they passed it on to one child. So I don't see any carriers in here. So a carrier would be somebody who does not have the trait, who passed it on to someone who does have the trait. Let's look over here. I see two people who do not, but they have two children who do have the trait. So these two individuals are going to be carriers. So that tells me that this is a recessive disorder. What you can do with a pedigree, once you've identified, either you'll have to identify that it's a recessive disorder, or I will just tell you in the test question. Now I can figure out the genotypes of these individuals. I know this is a recessive disorder. I'm just going to use A. So this person has to be big A, little a, big A, little a. I don't know this individual because they don't have the trait. So they're either big A, or big A, little a, but I'm not sure. These two individuals do have this trait, so I know it's a recessive disorder, which means if they have the trait, they are homozygous recessive. That's the only way to have a recessive disease. So these two individuals have to be little a, little a. I can actually go ahead and fill that in for all of the individuals that have that trait. This individual right here does not have the trait. However, they do have an offspring that does, which means they have to have at least one little a. The only way that this person has the trait is if they get one little a from dad and one little a from mom. So I know that her genetic genotype is big A, little a. Down here, we have two people that do not have the trait, so they have at least one big A. However, they have an offspring that has a little a, little a. So they each have to have a, little a, carriers here that have passed the trait on to an offspring. This one here we know has a big A, but we don't know the second one because we don't have any more information. So you'll likely see at least one or two multiple choice questions where I ask you about a specific individual and ask you either to give me their genotype on a pedigree or what their chance of passing on a specific genetic trait is to their offspring. So this is actually an example of a dominant disorder. There are some distinctive characteristics of dominant disorders that you can see. And so here, one is that if a child has it, the parent always has it. So here we can see a child with parents that have the trait, child with parents that have the trait, child with parents that have the trait. So a dominant disorder is going to show up in every generation. Whereas the recessives, you can see skip generations. This parent had it, it skipped this child, but it showed up again in this generation. So it's kind of skipping from here to here. Another characteristic of a dominant disorder is that about 1/2 of the children are affected. So just like we saw in the Huntington's pedigree or Huntington's Punnett square, there's a 50% chance of passing these traits on to the offspring. So here we've got 4 children and 2 out of 4 are affected. Here we've got 2 children, 1 out of 2 is affected. Here we've got 2 children, 1 out of 2 is affected. So this is not perfect, but it's a good way to kind of distinguish dominant from recessive. So here we know that this is a dominant disorder, which means that they could be either big A, or big A, little a to have the disorder. So what we can do is we can go to all the people who do not have the disorder, and we can write in their genotypes. We know that all the people who do not have, we can say this is some generic dominant disorder, are going to be little a, because they do not show the disorder. So somebody who is normal would be little a. And then we can try to fill in some of these dominant individuals. So in order for this person to have gotten 2 little a's, they would have gotten 1 little a from dad here, and then they would have gotten 1 little a from mom. So this individual has to be big A, little a. Same thing for here because they have 2 offspring without it, so this person has to be big A, little a. These children also have to be big A, little a because they can only get little a's from one parent. So this person has the trait because they got the big A from dad, but mom can only give little a. So these have to also be heterozygous. Same thing over here. This girl got her big A trait from mom, but dad would only have been able to pass on a little a, so she has to be heterozygous. This individual we actually can't decide without more information because they could get big A from both parents, or they could have gotten a big A and a little a from either parent. So this one is big A, or big A, little a. We wouldn't be able to figure this one out without more information.